Good morning, children, and welcome back to the regular classes, right? Okay, today we are going to do biotechnology principles and processes. What is biotechnology, children? Yes, it is the process of using organisms to get products out of them. You might have heard of genetically modified organisms. Yes, so that is what we are talking about. Okay, so biotechnology is the technique of using organisms or their parts, not the entire organism, even their cells can be used, or enzymes for production or for products and processes useful to humans. Right, and European Federation of Biotechnology, EFB. What is EFB? European Federation of Biotechnology defines biotechnology as the integration of natural science and organisms or their cells or their parts, etc. Okay, what is biotechnology? Again, it is the integration of natural science and organisms and molecular analogues for products and services. Once again, children, it is the integration of natural science and organisms and molecular analogues for products and services. Okay, now what does biotechnology process involve? First, it has two main steps. The first step is, that is what is GMF, genetically modified organism, GMO or GMF, whatever it is. GMO. How do we do the GMO? We remove an undesirable gene from the genome or the chromosomes and incorporate a desirable gene into its genome. Okay, this is the first step in biotechnology, which is the genetic engineering. The very first step is genetic engineering, which is altering the DNA of the organism by incorporating a desired gene from some other organism. That is the first step. The second step is providing an ambience. It has to get incorporated into the organisms, right? So we have to provide the proper conditions for it to get incorporated and also grow and replicate within the organism. So that is the second step. So providing proper ambience or conditions which is necessary for its growth. Okay, these are the two steps. First step is genetic engineering, where we alter the genes. We alter the genes by incorporating a desirable gene. Second step is maintaining of sterile ambience in chemical engineering process to enable the growth of only the desired cell or cell in large quantities. Okay, that is the second step. Clear? So what are the things done by using biotechnology? It is synthesis and using of a gene, preparation of DNA vaccines. Okay, now you know corona vaccine they've been trying to produce a RNA vaccine or corona vaccine for corona disease, COVID-19. Correcting a defective gene. Okay, if there is a defective gene in our chromosome, you have studied about chromosomal diseases and aberrations. Okay, so that can be corrected. That is, we can remove that and we can put a desired gene or corrected gene we can replace there. Okay, and also microbe mediated processes, which I've already studied in the previous lesson, which is making curd, bread, wine, etc. using microbes. Okay. Then in vitro fertilization, which is test tube baby programs. Okay, all these are the techniques used in, or the techniques done using biotechnology. Okay, now, so what we're talking about is we are removing a gene, a part of the genome, and replacing a desired gene over there. Okay, now how is it possible? First we have to cut a gene, to remove a gene and to replace a gene, we have to remove the DNA from the cell. Okay, so that is the very first process, very first step in the DNA biotechnology or genetic engineering process. Okay, so first step is removing or isolating a DNA from the organism, any organism, whether a plant or an animal or a bacterial, any cell, fungus, any cell. Okay, to remove. How to remove that, children? Children, you should also know the difference between sexual reproduction, asexual reproduction, hybridization, and recombinant technology. Okay, you know that asexual reproduction, by asexual reproduction, you get the next generation which are of the same type. Okay, we get pure lines, of course, right? That means asexual reproduction doesn't give rise to variations unless mutation happens. Okay, usually there is, it will produce the same kind of organism, providing less chance to variation. Whereas sexual reproduction also produces the same two parents, right? Sexual reproduction, there is involvement of two parents, and the new individual is having both the parents' characters, and also shows mutation as well as variation. Clear? That is the variation. Variation is there in the sexual reproduction. You might also know about hybridization. Now, what is hybridization, children? It is the process of crossing two plants or animals also, right? Two plants or animals with our favorite desired characters or desired traits. So we select two plants or two animals belonging to the same genus or same family, whatever it is, and we cross them. So that is what is hybridization, right? And we select the organism, the two plants or two animals having the best qualities in them, the best qualities. Okay, and the new variety, in the next F1 generation will have both the parents, both desired and undesirable characters. Yes, the parents, both desirable and undesirable characters. But recombination technology involves the creation of individuals with desired traits only. Okay, that is the most important point. We make the individual of our desire, what we desire, that we incorporate, that genes we incorporate into the organism. Okay, so also you should know, children, when we put this recombinant DNA, what is a recombinant DNA? A small piece of segment of DNA got from another organism and incorporated into another organism. That is called as the recombinant DNA, right? If a recombinant DNA is just introduced into a cell, just put inside the cell, will it get incorporated? Will it get replicated? Never, right? It has to get incorporated into the genome or the chromosome of the individual. Then only when the chromosome replicate, when the individual's chromosomes replicate, this recombinant DNA also will replicate along with them. Okay, now when this replicate and form many copies, right? How much time it takes for the cell to replicate, that many number of copies of this recombinant DNA will be formed. Okay, which is now called as clones, CLONES, or the method is called as cloning. Okay, right. So that is the difference between all these things. And the very first instance of recombination technology was done by Stanley Cohen and Boyer. Remember these two scientists, they will be asking the contribution of these two scientists, Cohen and Boyer. Now what they did was, children, you should also know, E. coli or bacteria. Bacteria usually do not have a nuclear membrane. They have a place called as where all these nuclear membrane, DNA and everything is present, is called as nucleoid. Yes, nucleoid. Okay, so besides this regular DNA, they also have a circular DNA which is called as plasmid. What is the name? What is the kind of name of that DNA called? Plasmid. So it is an extra chromosomal, autonomously replicating. It can replicate by its own. It does not depend. Its replication doesn't depend on the actual DNA inside the nucleoid. Okay, it is self-governing. It is an autonomous, self-replicating, circular, extra chromosomal DNA. It is extra chromosomal DNA. And children, also this plasmid may be in some bacteria, some E. coli, there may be more than 100 or 200 number of plasmids in the cell, which means it has its own replicating rate. It can replicate the one cell has 200 means you can imagine, right? So we can use this plasmid as a vector to transmit our desired genes into different organisms. Okay, this plasmid is a very best example of a vector. Okay, right. So what was the very first instance of recombinant technology done? It was, who was the two persons? It was Cohen and Boyer. And what did they do? They cut out a segment of DNA containing antibiotic resistant gene. Cutting out a segment of DNA which had the antibiotic resistant gene in it, and they incorporated this segment into Salmonella typhimurium, which is a bacterium which causes typhoid. Salmonella typhimurium is the name of the bacteria. Okay, in the plasmid of this Salmonella. And now actually the bacteria are not antibiotic resistant. Okay, now this bacteria, Salmonella bacteria, has become antibiotic resistant. Do you get me, children? Okay, what did they do? They took they cut out a segment of DNA which had what gene? The antibiotic resistant gene in it, and they incorporated into this plasmid of Salmonella. The Salmonella has become antibiotic resistant. Okay, usually the bacteria don't have antibiotic resistance. If any bacterial infection is there, and if you go to doctors, the doctors will prescribe a dose of antibiotics. You know that when we take antibiotics, the bacteria get killed. Okay, now the Salmonella has become antibiotic resistant. Do you get me, children? Okay, now in this plasmid, this will get replicated and form clones, for many copies of itself. Clone. We can use this vector to transfer into another organism, another bacteria also we can use this. Okay, children, now I've been telling all this while a small part of the gene has been cut. How is it possible to cut the gene, cut the segment of the DNA now? How is it possible? With what they cut? With the scissors or so, is it? Yes, there is an enzyme which is called as restriction endonuclease. What is that? Restriction endonuclease, which will cut the DNA, which cuts the DNA at specific points. Okay, we'll talk about that later. Let me come to the first step, okay, which is the isolation of DNA. Isolation of DNA. I told, isn't it, how to remove the DNA from the cell. So I told you we have to add different kinds of enzymes to remove what is that, cell wall. Cell wall is made up of cellulose. So if you add cellulase, the cellulose get digested. Chitinase for fungi. In the case of fungus, we can add chitinase to digest the chitin of the fungi. Okay, and there will be many kinds of biomolecules inside the cell, like lipids, proteins, RNA, DNA, and whatnot. So many biomolecules are there, right? To digest all these molecules, their respective enzymes can be added, like lipases, which digest fats, proteases, which digest proteins, RNases, which digest RNA. Like that, various kinds of enzymes can be added, and all the molecules can be digested. Clear? And what is left now? Only the DNA is left inside the cell, or now DNA is only left now, right? Which can be separated as what, thread-like structures by using chilled ethanol. What should we add last? Chilled ethanol. If you add to this the cells, now it will precipitate. The DNA will precipitate as thread-like structures. Okay, that is what you have to. That is how you isolate DNA from a cell, any cell, from animal or plant or bacterial cell. Clear, children? Okay.